Will be at the door, I guess.
Oh, thank you, Ken. That's great. To start now. We're all set? No. Test, test. Okay. We're going to start the panel on the AYAHUASCA research project. In 1993, as we mentioned last night, the three of us, Dennis McKenna, Jace, Callaway, and myself, went down to Manaus in Brazil, and along with collaborators in the Uniao de Vegetal syncretic ayahuasca church, we conducted a series of biomedical psychiatric studies on long-term members of the UDV who had used ayahuasca within the context of the UDV for at least 10 years. Our first presentation to discuss the project and to provide background leading up to it will be Dennis McKenna. Dennis is really one of the preeminent scholars in the area of ayahuasca investigations. He's made a number of critical contributions to the field and has really devoted much of the last 20 years to developing a very rigorous course of studies in this area. So it's a pleasure to introduce to you Dennis McKenna.
Well, thank you, Charlie. I'm going to give a somewhat different presentation than I usually give. Those of you who have seen lectures that I've given before, there's usually a lot of slides that you can't read and a lot of data that you can't interpret. And I figure that Jace and Charlie, between the two of them, will probably give you more data than you want. Or that you can possibly process. And so I'm going to leave the hard science to them, and I'm going to give you more of a personal sort of overview of this and talk about how I sort of stumbled into this ayahuasca research project, literally. And it's so, easy listening, it'll be a travelogue type thing. Before I get into the lecture, though, I did want to say something, call it, shameless commercial exploitation of indigenous property, intellectual property or something. But a number of people have asked me about these Pablo Amarengo t-shirts, and are they available? Well, yes, they are, but we only have a few copies here to sell. But if you want one, you can go to the maps table, and there's some order forms there. You can pick up an advance order form and either leave it at the maps table or fax it to the number on the order form. If we get enough orders by the 3rd of April, we'll do a production run and then send them to you in the mail. So I'm not going to say any more than that, but they are nice, and they're kind of an amazing pinnacle of the silkscreen process. I don't know how they managed to do this, but so if you want one, go to the maps table and they can tell you how to get one. In fact, I think there may be a few left. As I said last night, I guess a lot of my graduate school career was an attempt to understand or sort out some of the events that happened to my brother and I when we went to the Amazon in 1971. And that story is probably familiar to a lot of you, and in a lot of ways I've been trying to live out the resonance from that event for the rest of my life, or maybe live it down, I don't know. But in an attempt to do that, I decided to I went back to graduate school, and I was a graduate student at the University of British Columbia in 1981. When I first went there, I had been working happily on the genetics and enzymology of psilocybin biosynthesis, which was an interesting project in itself. And so I was more or less ensconced in the laboratory and enjoying that work. And my supervisor at the time, Dr. Neil Towers, invited me and another graduate student into the office one day and said, well, I have some extra money in this grant. How would you guys like to go to Peru for 6 months? And so we said, well, you know, our bags are packed. When do we leave, basically? And so I changed my direction and went to Peru to pursue a graduate thesis that kind of focused on the comparative ethnopharmacology between two Amazonian hallucinogens. We were using that outmoded term at that point, psychedelics, one of which was ayahuasca. If I could have the first slide. Well, that's interesting. No, I don't even need. No, I don't think so. But thanks for maybe. Do we have? It's jammed. Okay. Well, one of them was ayahuasca, which, as you know, is a combination, a beverage made from two plants: the Banisteriopsis caapi, or the vine Banisteriopsis caapi, which is itself called ayahuasca, and the admixture plant Chacruna, or Psychotria viridis, which contains dimethyltryptamine. And that is actually what supplies the hallucinogenic component, the psychedelic component, to the ayahuasca beverage. And one of the questions I wanted to investigate is, here you have two major Amazonian hallucinogens, and is there a similarity in their action? As we know, and as the research I did subsequently helped to demonstrate, the activity of ayahuasca depends on this combination of the Banisteriopsis alkaloids, these beta-carboline type alkaloids, and for the oral activation of dimethyltryptamine, and that's why it's orally active. There we go. Okay, so this just shows the two plants and the major chemical components in each. Okay, will this work? Now, the other plant that I was investigating, or plant complex I should say, was Varola. And Varola is a genus of trees in the nutmeg family that is very rich. The resinous part of the plant is rich in dimethyltryptamine, 5-methoxydimethyltryptamine, other types of psychoactive tryptamines. Now, in large parts of the Amazon, Varola resin is prepared as a snuff, and it's taken parenterally. It bypasses this monoamine oxidase deactivating system in the gut, and just goes directly into the brain in the snuff. However, some tribes, three notably, the Bora, the Witoto, and the Muinani, prepare an oral preparation from Varola resin. And they concentrate out the resin, and they concentrate it down and mix it with ashes, and they take these little boluses or pills, and they claim that that too is a very potent and short-acting psychedelic. So the question I was trying to ask was, you have here two botanically completely different plant complexes, but chemically similar. And so are the same mechanisms responsible for the action of ayahuasca also responsible for the actions of these Varola pastes? And so that was kind of the rationale for doing this study, and that's what brought myself and my colleague Don McCrae to the Amazon in 1981 to collect samples and try to sort this out. Well, this was my first view of the Amazon. We first went to the Amazonian river village of Pucallpa. This is the Rio Waijaga coming in on the on the plane, and this was really my first glimpse of the Amazon, or the first glimpse I'd had of it in 10 years, previously being there in 1971. Well, this was my second view of the Amazon. This is the main street of Pucallpa. They'd had some torrential rains the night we got there, and lights, camera, action. Anyway, they'd had quite a bit of rain, and so the streets were a little bit muddy. Am I doing this?
We need to have the light on for the video feed, so.
No.
For what video?
The video feed.
Oh, the video feed. Okay, well, can we get the slides on anyway? That would be good. I don't care if you have the light on, but the slides would really help move it forward.
Or the bulb just blew.
It's not my day.
We haven't stamped love, it'll take them a while, but
Keep going, man.
we saw it keep going.
Well, I don't even have anything to say without without the slides. I mean, this is what we were. This was supposed to be a travel log, so that means I don't have any notes or anything really substantive to say. Just corner. All right. Okay. So we got to Pucallpa, we got settled in, and a couple of the shaman of the ayahuasqueros that helped us most there are shown here. This is Don Fidel Mozambite on the left, and his teacher and colleague Don Juan, and they're standing there in front of the chacruna plants. And I had this contact actually from Terrence McKenna, my brother, and Kat had been to Pucallpa 4 years previously and had encountered these gentlemen, and so I had the contact from them. And they proved to be very cooperative and very helpful in providing the samples that we need to complete this study. This is Don Juan again, or Don Fidel, in front of his house outside Pucallpa, and his wife and children. Very interesting. Is that a jam, or is that... okay, I don't know. Another person I met in Pucallpa at that time, who later proved to be a very important player in this whole unfolding drama, was Pablo Amaringo Shunya, who is shown here playing the guitar. This was actually on a sort of a farewell send-off when I passed back through Pucallpa on my way home after completing the research. But he has proven to be a good friend, and I had no idea at the time I met him that he had been a former ayahuasquero. It wasn't until Eduardo Luna and I went back to the Amazon in '85 that his interesting background and his talents as an artist came to light. This is Pablo with his mother outside of his house. His mother has since passed on, but he's turned out to be. After an initial sojourn in Pucallpa, spending some time with Don Fidel and other ayahuasqueros, we moved on to Iquitos, which the main street of Iquitos is called the Prospero. It's, as you can see, it's a little more first world than Pucallpa. It's, at least the streets are paved, and it's a very busy river town. It's the largest Peruvian town on the Amazon in Peru. This is the famous neoclassical church in the main square of Iquitos. And the shanty town, the riverside town, which is much of which is floating and moves up and down in level. And this is Belim, is the name of it. Very busy, marketplace, a lot of stuff going on always. I mean, this is a contrast with your local mall. The reason that we went to Iquitos, that Iquitos was our next stop from Pucallpa, was because it was a jumping off point for the area that we needed to go to collect these Varroa samples. The ayahuasca, at least as we were investigating it, is kind of an urban mestizo phenomenon, so you really don't need to go into the jungle to access ayahuasca. You go to these urban centers in Iquitos, and there and around the outskirts you can find local ayahuasqueros. However, the quest for the Varroa paste was required a little more ambition, and the area that's now left. The use of Varroa as an orally active paste is kind of a disappearing phenomenon, really. Unlike ayahuasca, which is the tradition is thriving and alive and well in many parts of the Amazon, this use of the Varroa is much more obscure. And even the tribes that practiced it often remember it as something that their grandfathers knew about and something that they sort of know about, but they don't really do it anymore. But there were people that, as you'll see eventually, we found people that could do it. But the area where we did our collecting is about a day and a half's boat trip downriver from Iquitos, this is the Amazon here, to a town called Pebas. And from Pebas, you go up a river called the Yaguasyacu and to a town called Pucurquillo. I'm not sure what that means in Spanish. And at the confluence of the Yaguasyacu and the Rio Ampiyacu, we went up the Ampiyacu to another town called Brio Nuevo, which I believe does mean new shine. Just to orient you, here's La Chorrera, of the famous 1971 escapade fame, and the Colombian border, the border between Colombia and Peru, is the Rio Putumayo here. So the Ampiyacu is right there, and the Rio Ampiyacu is there, flows into the Yaguasyacu, which in turn flows into the Amazon. So that's the area where we did our collecting for these Varroas. Okay, the other, let me see. Just to show, well, this is the Yaguasyacu. This is what it looked like, and this is the mouth of the Rio Ampiyacu as we were making our way up this river to Brio Nuevo. This is what I looked like as we were going up. At least in my own mind, I thought we were on quite an adventure here. But between the fantasy and the reality, a couple of things happened. I lost the ponytail and the beard, and also the pretty assistant took off for other parts, and here we are at the main Maloca in Pucurquillo. These are my new assistants here, helping me collect some plant specimens. And here's what we came for. Not that guy there. The tree that he's standing in front of is Varroa Theodora, which is known locally as Kumala. All the Varroa varieties and related species are called Kumalas. There are many, many different types of Kumalas. This, you may recognize Terrence there. In an earlier incarnation, he was able to join us for this leg of the trip. He wasn't with us the whole time, but he was there when we went to this area. You know, he likes to be where the action is. So. This is what the Varroa resin looks like when you strip the bark off. It immediately oxidizes and turns bright red like that and sort of oozes out of the gash in the tree. And to make the ukue, you need to sacrifice a good-sized Varrola tree and strip the bark completely off of it. We contacted a number of people in this area who claimed to know something about how to prepare the Varrola paste, and in subsequent bioassays in the field and also laboratory assays, we found that most of them really didn't know what they were doing. They either prepared it from sources that were low in tryptamines, or some we didn't find any alkaloids at all. However, we did find a couple of older gentlemen who did know what they were doing. This is one of them. Don Milton was his name, and he claims to have learned the preparation of ukue, as this Varrola paste is called in Watoto, from his grandfather. And so it just shows him here stripping the bark, and then he soaks it in water and collects the resin and the water, and then it's a fairly simple process. Then he just cooks it down and adds some ashes into it, and you end up with this sort of bolus here. And if it reminds you of something obscene, the taste is pretty much like what it looks like there. However, the material that he made did prove to be active, and quite active at that. So the question of whether these oral Varrola preparations are orally active was pretty much settled. And later it turns out with this particular one, it turned out to be almost exclusively 5-methoxy DMT. Anyway, that was kind of the rationale for my comparative study, and I want to just summarize the main findings, which is between ayahuasca and Varrola. Ayahuasca, of course, has a very limited a very widespread ethnographic distribution, and in Varrola, the ethnographic distribution is extremely limited. And in ayahuasca, the plant families that are implicated are the Malpighiaceae, the family that Banisteriopsis is on, and the Rubiaceae, the family that Psychotria viridis, the Chacruna admixture plant, belongs to. In the Varrola, it's the Myristicaceae, the nutmeg family, and no other admixture plants of significance are used in the preparation of these Varrola pastes. Ayahuasca is an orally active beverage, and this is an orally active solid paste, or it's sometimes taken as a snuff. In ayahuasca, DMT is the active entheogen or psychedelic principle, and in Varrola, it's DMT, 5-methoxy DMT, and sometimes a whole sort of mix of different tryptamines, but those are the main two. In ayahuasca, there are high levels of the beta-carboline alkaloids, which are the MAO inhibitors that activate the DMT orally. However, in the Varrola, although it does contain beta-carbolines, or some specimens did, I found very low levels. The beta-carbolines were essentially trace constituents, and not enough in there to account for the oral activity, that not enough to be effective MAO inhibitors, and in fact the Varrola pastes were not. So there's kind of a mystery there. Ayahuasca beverages in the lab proved to be very potent MAO inhibitors. You could dilute these brews by orders of magnitude, by almost 10 orders of magnitude, and still get strong activity. And the Varrola pastes turned out to be quite so-so as MAO inhibitors, not particularly active. So what makes the Varrola pastes active is not really ever been resolved. I mean, there are theories, but we weren't able to figure it out. But they definitely are active. I'm just going to skip over that. This is my brother again, preparing to go back home. Those red seeds on the floor there are seeds of Varrola, which he was attempting to smuggle back into the States. So that we could try and cultivate them. Those of you from the DEA here can be reassured that we failed miserably with that particular project. They don't like to be taken out of their indigenous environment, so cultivating Varrola anywhere than the Amazon is is problematic, which is probably a good thing because in the Amazon it's one of the more common trees. There's actually hundreds of square miles that's basically nothing but Kumala. So now we go to 1985, and where I was invited by Dr. Luis Eduardo Luna here in one of his more informal moments to attend a conference in Bogota that he organized as part of the International Congress of the Americanists. He had organized a symposium on ayahuasca. And I came, and after we had the symposium, we went to his home in Florencia, where he grew up, where we're shown here, examining a local fungal specimen. And after that, we went to Iquitos and Pucallpa together. And he introduced me to some of the people that he had been working with in Pucallpa, and I introduced him in turn to Pablo Amaringo. And the rest, they formed an alliance, and eventually Eduardo and worked with Pablo to make his artwork and his paintings known to the world. Many of you are familiar with the book Ayahuasca Visions, which Eduardo Luna edited, and it features the work of Pablo Amaringo. But one of the most remarkable people that Eduardo introduced me to was the ayahuasquero that he had worked with for many years, Don Emilio Andrade, as he's shown here. And part of the, this is Don Emilio treating Eduardo's young son at the time, Semele, had some sort of minor ailment, colic or something, and Don Emilio was giving him a limpio of some kind. But part of what you understand when you go to this place was that you undergo a series of meetings with remarkable people, largely in this case remarkable men. I mean, most of the ayahuasqueros that we all of them were men, although there are women ayahuasqueros. But when you get to know these people a little better, you understand that although many of them are at an advanced age, they're usually extremely vigorous, they're extremely intelligent, they continue their practices through thick and thin, they live a hard life, and yet they seem to take to it very well. And it just seemed that they were quite remarkable people, physiologically and psychologically. And that was really the genesis of the notion that we should do a biomedical study on ayahuasca. We had the idea that if you could go in there and take some tissue samples or something, take some blood samples, do EEG, do something, you might learn something, because it seemed that their frequent use of ayahuasca was not far from being a harmful thing, was actually giving them unusual levels of mental clarity and physical vigor and so on. But it seems so problematic to do that in this context, particularly in the context of mestizo shamanism in Peru, where witchcraft is an ever-present element, and the notion that you could come in and take blood samples and urine samples and this kind of thing. I mean, we felt that it was really logistically very difficult to do that. So we wrote up a paper, a proto-proposal on this, Eduardo and I did together, and we just sort of put it on the shelf, not really knowing where to go, thought it was a good idea. Well, this is just more, this is Don Jose Corral, who is another one of the remarkable ayahuasqueros that he introduced me to. We saw him as recently as a couple of years ago. He's still going strong at 93, has a full roster of patients, spends most of his days chopping trees, very strong man. Well, so again, we didn't know what to do about it. So flash forward again to 1991, when we were invited to a congress organized by the Uniao de Vegetal, this one of the Brazilian syncretic sects. And both Eduardo and I were invited to give papers on our research. And this turned out to be the genesis of the real project, because these people were very receptive. We discussed with them the notion of doing a biomedical study, and it turns out that they had already had that very much in mind, so they were very receptive to the idea. In addition, many of the members of the UDV were medical personnel themselves, medical doctors, psychologists, this sort of thing, so they were familiar with the practices of Western medicine, and they thought it was a wonderful idea. So we said, well, let's do it with them. So we went back, we revised the proposal, we wrote it up more formally, and we were able to get funding for this, primarily through Botanical Dimensions, which was a nonprofit organization that Kat Harrison and my brother and myself had established some years before, and also MAPS and the Hefter Institute eventually provided some of the support for this work. So after this 1991 conference, we went back and tried to get the funding. Here are some of the participants at the Congress in '91. We tried to get the funding. Here is the temple outside Sao Paulo where this Congress was held. This is the UDV Nucleo, as they're called, and outside Sao Paulo. And we went back in '93, and we had basically, at that point, funding, modest funding, but an international team of investigators assembled, and we had objectives. We were very modest objectives, actually, because there was basically no data, and so where there's no data, you want to answer the questions that you can in the constraints of time and budget that will allow. So the objectives of our study were very simple. We wanted to look at the acute and long-term effects of huasca in human subjects. We wanted to assess serotonergic functions in long-term users of huasca. In other words, was there a difference between users and non-users that could be easily identified, not to say it's good or bad, but just was it present compared to non-drinkers of ayahuasca of comparable age and socioeconomic status and so on. We wanted to do pharmacokinetics on huasca. We wanted to measure the alkaloids in plasma, and we wanted to do quantitative analysis of the alkaloid content of various teas and source plants. Pretty simple objectives, and that was what we were trying to do. So we went to Manaus in 1993, and Jace Callaway by that time was part of this project. Myself and Dr. Grob here, we found Dr. Grob most willing to be the principal investigator, and since he had the MD, he got to be the guy, and and we went, and this study was held at the Núcleo Calpari, one of the older temples in Manaus, one of the older centers for the UDV, and this is a picture of the outside of the temple. And the first thing they started out with, they had to do what they call a preparo, which is basically an industrial scale production of ayahuasca. This was a smaller scale than they usually do because they were preparing this special research batch of material for us to use in the study. So I just have a few pictures of the preparo. This is the chacruna. All of these plants were growing on the site of the Núcleo Calpari in Manaus. And so they did it, and this just shows first they harvest the vine, and then they have to beat it with sticks to break it up prior to boiling it. And when you do that, it oxidizes very readily and turns red. And they get a big pile. It gives you can see this bright yellow color is characteristic of the beta-carboline content. The beta-carbolines are fluorescent, and so they have a, so then they just load it up. And this is a group project, obviously. A lot of people get involved. The fellow on the right there with the black belt pouch is Glaucus, who was head of the medical division of the UDV and is an epidemiologist in his own profession, and he was the principal investigator of this on the Brazilian side. Really a fine gentleman. So they get these things lined up, and then they just go. They continue this throughout the night. The boiling process is about a 12 to 16-hour process, and they just run through. Of course, they have to test the batch periodically while it's being processed to be sure the quality is good. So it's quite a festive occasion. And then they filter it, and then that's Glaucus again testing looking at it to make sure it has the right color. You can actually tell a lot about the color. That's making some specimens here of the source plants for analysis later. Pouring some of the Huasca in again. All of these were shipped eventually to Finland, where Jace Callaway carried out the analysis of these brews. And this is the picture of the completed brew, and the rainbow sheen on the surface of the ayahuasca is a way to tell if it's a good quality because of these fluorescent beta-carboline alkaloids. So this one looks, pretty good, and that's my segment of this thing. I'm going to let Charlie and Jace go on from there with the real data. Thank you.
Okay, our next presenter who will discuss the biochemical findings is Jace Callaway, who's a professor in the Department of Pharmaceutical Sciences at the University of Kuopio in Finland. Jace is primarily responsible for the what I consider to be extremely important findings in regards to the long-term effects of ayahuasca on the serotonergic neurotransmitter system. This is a phenomena which we all believe has considerable implications for the field of psychopharmacology. So Jace will now share his work.
Thank you for that kind introduction, Charlie. Thank you, Ralph, and the other organizers for having me here. It's a great honor. I wish I were talking a little bit more about the subjective end of things. I think it's the more interesting part of this. But unfortunately, I need to bore you a bit with some facts and details. I hope you people in the back can see these slides. Could I have the first slide, please? And maybe at least that light off. For you people in the back, I'll just read this quote from the Yahe letters. This was Allen Ginsberg writing to William Burroughs saying, "I could hear retching and groaning as if I were someone else." And that's exactly true. It does feel like someone else is vomiting their guts out during this time. There's a depersonalization that's quite characteristic of this experience. If we could get this in focus, it's a rather detailed slide, but what we have here are the major alkaloids involved in ayahuasca. There's harmine. This is a strong inhibitor of monoamine oxidase A. It's an enzyme in the liver which metabolizes serotonin and dimethyltryptamine, among other things. Also at this dose in ayahuasca, I believe we're also inhibiting monoamine oxidase B, the whole spectrum of monoamine oxidase, not just A. Another important alkaloid is tetrahydroharmine. It's a weak inhibitor of MAO. But more importantly, it's a weak serotonin uptake inhibitor. Prozac, for example, is a strong serotonin uptake inhibitor. So this is playing at a little different receptor site than monoamine oxidase itself. Finally, dimethyltryptamine, which is common in most teas. This is binding to serotonin receptor sites. It's actually competing with serotonin during the ayahuasca experience. It's agonistic at 5-HT1A and antagonistic at 5-HT2 sites, and also binding at 5-HT1C. So 3 distinct receptor sites are receiving this, and these are essentially resulting in the visual effects and also deeper cognitive effects. One more slide of molecules to entertain you. And in the middle, I have serotonin here, and these are different tryptamines most of us are familiar with. Here's dimethyltryptamine, bufotenine, or 5-hydroxydimethyltryptamine. Here's 5-methoxydimethyltryptamine and 4-hydroxydimethyltryptamine, otherwise known as psilocin. You can see a distinct similarity between all of them. They're all tryptamines. They're all binding at the serotonin receptor. It's not important to really know about molecules to see these similarities, and that's the only reason I'm putting these up for you to see. Essentially, dimethyltryptamine, when monoamine oxidase activity is functional, results in no oral activity whatsoever. It can be smoked or snorted or insufflated and injected to get activity, although that's qualitatively different than if monoamine oxidase is inhibited. If the enzyme is inhibited, there's oral activity and profound psychoactivity. This is the basic mechanism that's going on. Honestly, I have no clue how these people stumbled onto this combination of plants. It's nothing short of a miracle, and any explanation belongs in mythology. There's no rational explanation why people could have stumbled upon this. A little more information on monoamine oxidase inhibitors. As I said earlier, there's monoamine oxidase A and B. These are isozymes, essentially just different variants of the same enzyme in the body, and they metabolize different chemicals. I've just listed a few specific inhibitors of monoamine oxidase A, chlorgeline, harmaline, and harmine, and monoamine oxidase B, deprenyl, most tricyclic antidepressants. And then there are a host of non-specific MAO inhibitors that just indiscriminately inhibit both A and B. A cartoon of a neuron, as best as I can draw it. This is the presynaptic neuron and the postsynaptic neuron, and the synaptic cleft in the middle. The serotonin uptake site on the presynaptic neuron. This is where Prozac binds. There's also an endogenous compound that we make ourselves called pinealine that binds here, which is very similar to tetrahydroharmine, which is found in the tea. As the neuron takes tryptophan in, it produces serotonin, which is stored in these vesicles. And when the nerve impulse comes down, we release serotonin, which binds to the postsynaptic site. Any extra serotonin is binding back over here on autoreceptors to tell the neuron, stop firing, there's enough serotonin, and that does it. Other stuff will be recycled and repackaged for later release. This is the basic function of a neuron. Dimethyltryptamine is basically coming in here and binding onto these different sites and causing just a little different action than normal serotonin. For most of us. Thank you. These are pictures of samples that were analyzed. We had 2 types of banisteriopsiscopy. It was mentioned earlier that there are many known by the indigenous people. The UDV recognizes 2. A knobby one, which is stronger and causes a lot of vomiting. They consider that to be force. This is called kaupari. And another variety of banisteriopsiscopy, which is milder, called tukunaka. I analyzed both of these, and the kaupari has much more higher alkaloid content than the tukunaka. So this explains the difference in force. It's, in my opinion, it's just a difference in alkaloid content. I have to say here, these alkaloids are really just associated with the effect. We really don't know what the hell is going on, you know, at the deeper level of psyche. And again, I wish I were talking about this, but it's completely subjective and beyond the scope. I'll also mention that I've analyzed the leaves of the tukunaka, and you can see that there are very high alkaloid content in the leaves. And I mentioned this to the people at the UDV because they essentially throw these on the compost heap. I said, hey, you guys, there's more alkaloid here. And they said, yes, yes, we know that, but it gives the tea a bad taste. Also in this particular batch of leaves, the Psychotria viridis, which is called chacruna, was about 6 milligrams of DMT per gram of dried leaves. I should also mention that the Banisteriopsis copy is called mariri. That's the name that the people use there. And the Psychotria viridis is chacruna. Just to show you the variation in alkaloid content of the Psychotria viridis, I had about 35 samples of leaves that I looked at one particular time. This scale is milligrams of DMT per gram of dried leaf. It goes up to about 17 milligrams, and most of them were in the range of about 8 milligrams per gram of dried leaf, and I had a couple with no detectable amounts of DMT whatsoever, which was really surprising to me, because these leaves from these particular samples were thought to be active. They rated them on a scale of activity of force and light, light being essentially the component from the chacruna or the Psychotria viridis. And it doesn't really seem to me that the DMT is so important. In fact, the UDV seem to think that the visions that come from the experience are a bit of a distraction that one tries to burn through as quickly as possible to get to a higher state of mind to deal with this effect, to work out their problems. This was a surprise to me because from the literature I'd read previously, I thought it was all about visions, and indeed it is not. This was a study done with injected DMT in 1976 by Gillen et al. Just to give you some idea of the different parameters that they were measuring. Everybody had visual hallucinations, 100% of the volunteers, 15 normal volunteers. Hallucinations with eyes closed, and then other things. But particularly interesting, and I think Jeremy Narby touched on this a little bit, there were about 27% of the people who were hallucinating real things. Well, that's another story. I also analyzed, I don't know, about probably several hundred different tea samples to date, but on this slide there's just about 20 of them. There were 20 from the UDV. The DMT levels ranged from 0 to about 6 milligrams per milliliter of tea. There's a good amount of tetrahydroharmine most of the time. Harmaline, trace amounts. Harmine was quite heavy. I had one tea sample from the Barquinha, very high levels of DMT. I had one sample from Santo Daime at this time, again, very high levels of DMT, and fairly respectable levels of the other alkaloids. So again, it's not necessary or sufficient that ayahuasca teas have DMT in them. They're coincidental. They happen a lot, but it's not the main event. And earlier, Eduardo Luna was talking about a plant, Calliandra pentandra, and it does indeed contain tetrahydroharmine. And this is an admixture. So I'm beginning to think now that tetrahydroharmine is probably as important or at least as interesting as DMT, although the effects are quite difficult to characterize. They're not visual, they're more temporal in a way. Now, we started this study. This was our first man in space. We administered the tea. By the way, we had no idea of the alkaloid content. It was deemed to be appropriate for the study, but we had no idea really what dose to give people. We settled on 2 milliliters of tea per kilogram of body weight. So this man is taking his dose. We didn't know many other things. For example, they always drink the tea with the right hand, and that's where we had the catheter. So subsequently we put it in the left arm to make it a little more easy to bend the elbow to get the tea down. This is always administered by a mestre, and we kept in keeping with their beliefs and customs throughout the study, essentially. Luckily, the UDV sit down almost entirely throughout the time. This made it very convenient to take blood samples on his schedule. So he drinks the tea, we go right away for the first blood sample.
Got to get the baseline.
Yeah, we've got to get the baseline as quickly as possible, no kidding. So that was the first day we just did one. At the high point of the study, we were doing 3 a day, staggered every 15 minutes. This was a circus by any measure. So this guy has had his, and he's getting his temperature taken now, already had his blood sample. This Mestre Glaucus Brito, our medical doctor at the site, is administering the tea here. And still, here we are, cuffed to the right hand, and he just thinks, okay, I'm going to do it with the left hand. And they are very rigid, but this is an important thing, and he just decided, I'm going to do it with the left hand. It's no big deal. He wasn't punished or anything like that. Just to give you an idea what our lab setup was like, this is the table where most of the things happen in the nucleo, and we essentially turned it into a small medical hospital. Here's our victim here while we're processing all kinds of EEG stuff. You'll notice a stereo here. It's very nice to have a little music, as some of you probably know during such an experience. And this music is carefully selected before every session. It's a very serious thing for these people. Oh, where are we now? This is essentially body weight tea administered. The average body weight was 74 kilos. Everybody had about 150 mils of tea, plus or minus 20, and these are just the different doses of the various alkaloids. I'll not spend much more time on that. Now, something that did come out that was quite interesting, and this is essentially new, we're looking all the time in this data, a huge amount of data to sift through. We found basically 2 groups of people in a small study of 15. These are all men, by the way. Essentially people metabolizing this fast and slow. And I use quotes because these are very relative terms. The maximum concentration of harmine in the fast metabolizers was about 60 nanograms per mil, and in the slow metabolizers 152 nanograms per mil. So this shows a very clear difference between people. That some people are going to handle this a little different than others simply because they metabolize these alkaloids at different rates. Now the times that these Tmax, the times of these things were not so different, but the total concentration that was floating around in the blood at the time we took the sample varied significantly between these 2 groups. There were about 6 people in the fast group and about 9 in the slow group, for whatever that's worth. I still don't know what this is about. There was some difference in weight. The fast ones were a little lighter than the slow ones, but it was not statistically significant difference. This is harmine. This graph is the blood concentration. This is 500 minutes on the end. The lower one is the slow metabolizer. The lower one is the fast metabolizers. The higher one is the slow metabolizers. So harmine is staying around in the blood much longer in the slow metabolizers. This is dimethyltryptamine, again in the slow metabolizers, higher concentration. Fast metabolizers, low concentration. And you'll notice these are averaged. I don't have error bars on here. You'll notice that there are these bumps like this. They're not nice curves. This was disturbing at first. I thought maybe the analysis wasn't working, but it turns out that these alkaloids are recycled through the liver and the bile. They're reabsorbed from the large intestines and taken back through the bile and sent again through the system. This is called enterohepatic recirculation, and it happens with some other drugs too. The other compound, tetrahydroharmine, this is the slow metabolizer and the fast metabolizer. Really not so much difference, particularly as you come out here on the end. So tetrahydroharmine is really not playing into this monoamine oxidase metabolic pathway. It's something a little different, and the difference between fast and slow don't really apply as critically to this alkaloid. Something that was the most interesting and unexpected finding was that the blood platelets of the people who had used this tea regularly over 10 years or more had higher receptor sites, a higher density of receptor sites of the serotonin uptake site. Now, in our blood, we have platelets, and on the platelets are receptor sites for serotonin, just identical to what we have in the brain. So this is a very convenient sample. We can just get blood samples rather than brain samples from these nice people, and and we can tell that there's some difference between people who have never had this tea and people who have had this tea over quite some time. Now, these blood samples were taken 1 week after the last time they had had ayahuasca. There were no alkaloids on board in these people. They were completely drug-free. This is just reflecting a change, a very fundamental change in the serotonin receptor population. It's also interesting because typically drugs will decrease this parameter. This, to my knowledge, is the only substance that increases this parameter. I was a bit at a loss to explain this at one time. We published these results in 1994 in the Journal of Psychopharmacology, and it seems that the tea acts as some sort of a challenge. There's a huge amount of serotonin coming up in the system when monoamine oxidase is inhibited. So you not only have the alkaloids of the tea in their body, there's also your own serotonin is increasing at very high levels and having effects. The nausea comes from this, the diarrhea comes from this as well, and also a special kind of state of mind when all the serotonin receptors in the brain are completely activated by one's own serotonin. And DMT is desperately trying to compete with this and successful to some extent, finding some receptor sites as well. This is a relatively long-lasting change. By that I mean it's about a month. It takes a month to induce it and about a month for it to go away. Very, very interesting phenomena here, because this is exactly what an antidepressant should do, but they don't. So what I suggest here is that yes, I'm going to suggest it. There are some components of the ayahuasca or huasca tea that are apparently useful, perhaps in treating depression and certainly drug dependence such as alcoholism. And I say the latter because the UDV is a group of about 8,000 people now. To my knowledge, no one drinks alcohol in that group. No one smokes tobacco. Charlie will present some data to show that quite a few people had drug problems before they even started. So in my mind, this is an ideal candidate for addiction interrupter, not to mention treatment of intractable depression. And there's plenty of anecdotal evidence to support these speculations of mine, these wild speculations, I might add. And something else, I had to take this a little bit further, this serotonin uptake thing. We do a bit of this work at the University of Kuopio, particularly with violent alcoholics. We're finding in their brain there's a decreased amount of serotonin uptake sites in the brains of violent alcoholics. So some of the clinicians were rather interested in this finding because here is a drug that could potentially increase serotonin uptake sites. In alcoholics, I don't know. In normal people, yes, most certainly. So let me just show you a couple more slides and then I'll be done. Well, in the top here is the brain scan of a normal person. Thank you very much. And what's lit up here are serotonin uptake sites. And particularly this little bundle is characteristic of controls. Now this is a normal brain. This is not my brain. This is a this is a normal, healthy, finished brain. And now down here is the brain of a violent alcoholic, and there is a substantial decrease. Am I getting off, or is this moving around a lot? I just get so excited talking about ayahuasca, really. Oh yes. You'll notice this little spot is absent in violent alcoholics, and this finding has been published. It is real. There's no question about it. So the obvious question was, will we see such a thing in normal volunteers, or should I say a normal volunteer, or maybe let's just leave the normal out of it completely, a volunteer. So this particular volunteer had a brain scan, and unfortunately this is, could we turn that light out there, please? That helps a little bit. Yeah, September 6th, this is the brain scan before, and then October 18, another brain scan after a course of what I would call pharmahuasca in honor of Jonathan Ott, who coined that term, several times a week for during this time from September to October. And what we see here is a substantial increase in serotonin uptake activity in the brain is measured by this technique, and you can't see it very well, certainly not in the back, but this bundle is also increased as well. Here it's not so apparent. This almost looks like a violent alcoholic brain. I assure you I'm not violent nor alcoholic, but I do think this kind of runs in my family, so this may be a genetic sort of thing. And so N of 1, this is not really a study, it's preliminary. All kinds of preliminary studies get all kinds of things, but to me this is encouraging, and it fits in rather well with what was observed in the blood platelets. There is some kind of apparent effect, not just this short-term visual thing that goes on, but some long-lasting effect from a regular consumption of ayahuasca, huasca, or these kind of teas. And now here's Charlie.
Okay, I'll provide the final presentation on our study. Okay. We could cut the overhead light. Now, Jace told me this slide here was from the magic eye.
Yep.
So can anyone make out what it is? I can't.
Charlie, you want this light out?
Yeah, take it out for a second.
Well, actually, we need that light on for the video feed. For the video.
All right, well, Jace will share the answer with you later. Okay. Okay, great. Is there an autofocus? Okay, so essentially this was the structure for the research study we did in June and July of 1993 in Manaus, Brazil, with subjects from the Uniao de Vegetal who volunteered. We used 15 volunteer subjects with at least 10 years of ayahuasca experience and 15 matched controls who had never taken ayahuasca. And you can see here, Jace has already talked about what I consider to be some of the more fascinating findings, which is the receptor binding work and the possibility that we may indeed have the basis for an entirely new psychopharmacologic model for treatment of mood dysregulation. Jace also mentioned the pharmacokinetic assessment, and this is basically what else we did: baseline medical exams, medical workups in both UDV subjects and controls, structured psychiatric diagnostic interviews, neuropsychological testing, personality testing, the phenomenological assessments, semi-structured life story interviews, and all of these were the baseline medical and the diagnostic, the neuropsych, the personality, and these were all done with both groups. A phenomenological assessment was done with our experimental subjects after the experimental session, and the acute medical parameters, as Jace showed you some slides of our subjects undergoing procedures. Basically, we tried to match them as well as we could. They averaged in the mid-30s range, 35, 34, 36 years. We matched them along a lot of parameters, except we found that education and socioeconomic status, interestingly, our UDV subjects were asked to bring in an associate of theirs, a friend of theirs, a colleague of theirs who had never drank the tea in an effort to match them as best we could for lifestyle, diet, etc. And they tended to bring in individuals who had somewhat higher educational level, which was curious. Here you see we measured temperature. Show you a few slides here. Our first subject again. Pupillary diameter. Here we had Dr. Andrade from the University of Amazonas Medical School, the Department of Internal Medicine. We ran EKGs with all of our subjects. I might mention that, as we talked about last night, these were preliminary investigations. There's clear need for follow-up, and there were some holes in the data set we collected. One of the problems was with the EKGs, because of the extremely high humidity in Manaus. This material had preliminary readings and were then put in a desk drawer, and unfortunately the paper decomposed. So this is one area that needs to be pursued further. I can mention a little bit later some of the preliminary EKG findings. Okay, here we have some of the acute medical parameters. On top, I think this is heart rate. I don't know if we could move it down a tad, but you can see that
No, it's temperature.
that's pupillary diameter. Okay, so you can see it about going up at about 1 hour. It peaks and maintains a dilatation. Here we have the respiratory rate. And down here, the oral temperature, very slight elevation over the course of the 4 hours worth of measurements. It only went up by approximately 0.3 degrees Celsius. And this may actually be an artifact of the setting we were working within and the timing. We started our sessions uniformly at 9 o'clock in the morning, and by 12 o'clock and 1 o'clock, the ambient temperature had become extremely hot. So in fact, this may more likely reflect the ambient environment. Here we have the heart rate. You see an initial elevation, even before the onset of subjective effects. This is felt to be a rapid effect of the MAOI on norepinephrine, causing slight elevation of heart rate, and then equilibration within the system and a drop in heart rate. And you can see that after couple hours or so, the heart rate is down below the baseline. Blood pressure, slight elevation early on, and then coming down to baseline and even a little below. Here we have systolic blood pressure, and here diastolic. We had approximately about 10 millimeters of mercury rise acutely, and then coming down quite a bit. So there did not seem to be severe risk for hypertensive response, at least in these subjects. We did, blood was drawn approximately every 20 minutes. Blood was later assayed for pharmacokinetics as well as neuroendocrine secretion. And we looked at 3 hormones. We looked at growth hormone, prolactin, and cortisol, and we found essentially that growth hormone peaked acutely at around 90 minutes and came down. Prolactin peaked at about 120 minutes and came down. cortisol peaked at about 60 minutes and then came down. By 4 hours out, it was pretty much back down to baseline. With the MDMA work we've done similar neuroendocrine assays, and we've seen a similar phenomena that during the period of acute subjective effect, there is increased secretion of hormonal factors which return to baseline as the subjective effect wanes. I may also mention that the MDMA secretions were generally quite a bit higher than what we found with the ayahuasca subjects. Oh, occasionally let our guys rest, but as you can tell, not for long. I might mention that our subjects were quite stoic. They endured a great deal and didn't flinch. However, we had a hell of a time with controls. for the platelet serotonin receptor studies that Jace was running, he was trying to get about 8 tubes of blood on each subject, and we had a couple of guys who saw the trays lined up with tubes and were told it was all for them, and they just took off. And last we saw, they were headed out into the forest somewhere. We had collaborators, psychiatrists and psychologists from several medical schools in Brazil, particularly from the Escola Paulista Medicina in Sao Paulo and the University of Rio de Janeiro Medical School, and we conducted a variety of psychiatric measures, which I'm going to share with you now. Structured diagnostic interviews demonstrated with controls, for the most part, pretty clean psychiatric histories. For our UDV subjects, on the other hand, quite a bit of alcohol abuse, drug abuse, as well as some depression and anxiety. Whenever you're concerned about a psychoactive substance and you want to do a risk assessment, you have to consider the issue of, could it possibly be neurotoxic? If so, how might that manifest? So you want to look to see if there's any deterioration in cognitive function or neuropsychological function. So we utilize with both the subject and the control groups, and remember this is baseline, not under the influence. And it was following a week's hiatus for our UDV subjects from the last time they drank the tea. We utilized the WHO UCLA auditory verbal learning test as a pretty simple and efficient means by which to assess whether or not there was any measurable neuropsychological damage in our ayahuasca using subjects. And what we found, not really to our surprise, because we had been regaled with stories by all the UDV members who we had met about how ayahuasca had really improved their powers of concentration, improved their memories. We found that not only was there no evidence of neuropsychological deterioration in our ayahuasca using subjects, but in fact on a number of several measures they scored quite a bit higher, significantly so here, than our non-ayahuasca using controls. So we felt this was very positive reassurance that the substance was not toxic. And if we're to listen to the long-term users, there might even be positive effects over time. We wanted to measure baseline personality assessments in both the ayahuasca group and the non-ayahuasca controls. We utilized the Cloninger tridimensional personality questionnaire, which consists of 3 domains. The first one, comparing the two groups, is reward dependence. We found no significant differences. However, in the second group, which was novelty seeking, we found that in a few different areas there was a significant difference, specifically here that our UDV subjects displayed far more stoic rigidity as opposed to our controls who had more exploratory excitability. And down here, our UDV subjects were more regimented as opposed to disorderly. Here we have the other domain, which is harm avoidance, and we found that the UDV subjects had far more confidence as opposed to fear of uncertainty that the controls had. Also, they were more gregarious, more social, and far less shy with strangers. Now, this may have been the long-term cumulative effects of taking ayahuasca within a religious ceremonial structure, or it may have been these may be factors almost pre-selecting what individuals will gravitate to take advantage of these opportunities that are available in Brazil. I'll also mention, as I did say last night that in Brazil, since 1987, the use of ayahuasca within the context of religious practice has been legal, fully sanctioned by the government. And here, just summing up what we found with the personality questionnaire, we found that our ayahuasca-using subjects, that the low novelty seeking that they scored on was associated with more reflectiveness, somewhat rigid, loyal, stoic, slow-tempered, frugal, orderly, persistent, high social desirability and emotional maturity, the low harm avoidance associated with confidence, relaxed, optimistic, carefree, uninhibited, outgoing, and energetic, and the association of the two, the low novelty seeking with the low harm avoidance, associated with hyperthymia, which means positive mood, persistent positive mood, cheerfulness, stubbornness, and some overconfidence. To assess the phenomenology of the experience, we utilized Rick Strassman's hallucinogen rating scale. All of the ayahuasca subjects were asked to fill out the long hallucinogen rating scale questionnaire 2 hours following the closure of their experimental session. And Strassman's scale, which he developed within the context of his FDA-approved intravenous dimethyltryptamine studies at the University of New Mexico in the early 1990s, is a 1 to 5 scale looking at intensity, form, feeling, perception, and down the line. And what we found basically was that compared to the intravenous DMT, that ayahuasca induced a relatively mild experience. So there's some significant differences then, depending on how the tryptamines are administered, whether they are activated orally or whether they're taken through smoking or injection. I might also say that Strassman, with his intravenous DMT studies, found far more blood pressure lability with his subjects than we did with our ayahuasca-using subjects. One of the more interesting aspects of the study, personally for me, was doing extended life story, open-ended interviews with each of our 15 UDV subjects. Essentially, I sat down with them and asked them essentially the question of, tell me about your life from before the time you first drank the tea, tell me about your first experience with ayahuasca, and tell me how your life has changed. And from there we went. I got a great deal of information, and here's what I found. And we remember a total of 15 subjects. Prior to entering the UDV, 11 of these subjects reported a history of moderate to severe alcohol abuse. 5 of them had histories of alcohol-associated violence. So this is 1/3 of our sample did actually have histories of being violent alcoholics. And these were the same men who demonstrated very robust serotonin measures. Of course, this is at least 10 years after their last drink. 2 of the subjects had been imprisoned secondary to violence. 4 of them had history of cocaine and or amphetamine abuse, and 8 of them were addicted to smoking cigarettes. Within the context of the UDV, as Jace mentioned, no one drinks alcohol, no one takes drugs. I had an interesting little dialogue going with one of the men who I asked him if he had ever taken drugs prior to becoming a member of the UDV. He said, "Oh, I've taken all sorts of drugs. I've done them all. I've done heroin, I've done cocaine, I've done amphetamine, but I've done them all except one, the most dangerous of the lot. I've never done marijuana, thank God." So within the context of the UDV, cannabis is not a welcomed ally as it is in other contexts. So it's interesting to look at how different groups who have utilized this substance, ayahuasca, may differ on various points, such as whether or not cannabis would be an acceptable part of the experience. Additional ways in which our UDV subjects describe themselves included, this is prior to entry into the UDV, being impulsive, disrespectful, angry, aggressive, oppositional, rebellious, irresponsible, alienated, and unsuccessful. So as you can see, looking back at least, this is all retrospective of course, a better study would be prospective, but this was preliminary, but looking back, they identified themselves as having significant problems, many of them with I found with several of them, their first experience was an extraordinary one for them, and really set the stage for what became their many-year relationship with the church, transforming their lives. But looking at the content of their first experience and the visions they had, one of them described, "I had a vision of myself in a car going to a party. There was a terrible accident, and I could see myself die." Another one said, "I was at a carnival on a carousel going around and around and around without ever stopping. I didn't know how to get off. I was very frightened." Third one said, "I could see where I was going with the life I was leading. I could see myself ending up in a hospital, in a prison, in a cemetery. I saw myself," and then the final one, "I saw myself arrested and taken to prison. They were going to execute me for a horrible crime I had committed." So this is the first experience. These four men had severely dysfunctional lives, a lot of alcohol abuse, violence, different kinds of drug abuse, and they had a terrifying vision. They could see, in a sense symbolically, where they were headed with the life they were leading, and then they saw that they were at a crossroad. They could continue going down that way, and here was going to be the outcome, or they could take the opportunity that was being handed to them and transform and change. Many of them also, in the throes of this really scary experience, had, for what to us might be the uncanny experience, but for them entirely understandable within the context of their structure, they experienced encountering the Maestri, the founder of the church, Maestri Gabriel, who Eduardo Luna talked with us about early this morning. And encountering the Maestri led to some additional very intriguing sets of visions. One of them said, "I saw these horrible, ugly animals. They attacked me. My body was disassembled. Different parts were lying all over the ground. Then I saw the Maestri. He told me what I would need to do to put all my body parts back together." Remember, this is traditional shamanic phenomenology, the dismemberment of the body, and then somehow in the experience, the rememberment as, in a sense, the rebirth from the symbolic death. A second one said, "I was in a canoe, out of control, going down the river. I thought I would die, but then I saw the Maestri in a canoe in front of me. I knew that as long as I stayed with the Maestri, I was safe." And essentially, that's how the church is structured. The Maestri, the memory of the Maestri, the founder, and the active Maestris who stand in for the original Maestri provide that safe vessel for the members. Another one said, "I ran through the forest." Oh, actually, this was a young man who actually did ran through the forest. He got so frightened with his first session, he had such horrific visions of violence and destruction and dismemberment that he just got up and took off. He said, "I ran through the forest terrified that I was going to die." But then he was just stopped in his tracks because, so he tells us, "I saw the Maestri. He looked at me. I was bathed in his light," which they described as kind of a green light. "I knew I would be okay." Now, they talked very clearly about the importance of the ritual structure. They had very clear, very strong opinions, and I'm just sharing with you what they told to me. Subjects say that they saw their use of ayahuasca as a catalyst in their psychological and moral evolution. However, they emphasized that it was not the ayahuasca alone that was responsible, but rather taking it within the context of their particular ritual structure. Rivalry between the different groups. Subjects were critical of other groups who use ayahuasca because they're critical that they're sometimes used in less controlled and less focused settings. One thing the UDV does well is control. This is somewhat problematic to some individuals who like a little more freedom, a little more expansive setting, but the UDV runs an extremely tight ship, and they might be criticized for perhaps holding back certain elements of experience, but that degree of control in many respects does optimize safety. Subjects describe the UDV as a vessel that enables them to safely navigate the often turbulent states of consciousness induced by ayahuasca ingestion. Subjects describe their church, the UDV, as their, quote, mother, their family, their house of friends, providing guidance and orientation and allowing them to walk, as they describe it, the straight path. Subjects emphasize the importance of Uniao, or union, of the plants and of the people. And finally, the subjects contended that without the structure of the UDV, the ayahuasca experience may be unpredictable and lead to an inflated sense of self. Within the house of the UDV, as they describe it, however, the Huasca-induced state is controlled and directed, quote, down the path of simplicity and humility. So that's essentially our life story interviews, and our other psychiatric and psychological workups were published in '96 in the Journal of Nervous and Mental Disease. Oh, for a little more on the life story interviews. They're a little out of sequence. Changes following entry into the UDV. They discontinued alcohol, cigarettes, drugs of abuse. So essentially, these are people, heavy alcohol histories, lots of different substances, entirely discontinued once they initiated this process of belonging to the church and taking part in ceremonial ayahuasca sessions at least twice monthly, the first and third Saturday nights of every month. They never take this sacrament outside of their ritual ceremonial structure. They described how in their lives they had undergone a radical restructuring of their daily conduct and orientation. And I'll give you some more quotes. One here says, I used to not care about anybody, but now I know about responsibility. Every day I work on being a good father, a good husband, a good friend, a good worker. I try to do what I can to help others. Another said, I have learned to be calmer, more self-confident, more accepting of others. I have gone through a transformation. And finally, this man said, I now know the importance of practicing good deeds, watching one's words, and having respect for nature. You know, it was a fascinating experience to sit with these men and hear about their terribly dysfunctional lives early on, how they had been alcoholic, violent, served time in prison, but sitting with them after at least 10 years as being involved in this process, these men were pillars of the community. They were highly successful out in the world and highly responsible in terms of their family relations and their interpersonal relations. So these men, we all felt, had truly undergone a transformation. Okay. This is a reminder. I was talking with Jace last night. How are we going to get a particular issue, how are we going to remember to bring it up? And he said, here, take this slide. So I'm not talking about, what?
And call me in the morning.
Call me in the morning. Okay.
But I'm not a doctor.
Right.
I mean a real doctor.
Okay. So anyway, what we're not talking about in this particular context is ayahuasca and a pill. Rather, what we're talking about is the fact that in our modern culture, many individuals are taking prescription medication. Many individuals take antidepressants, particularly in the last 10 years, selective serotonin reuptake inhibitors such as Prozac, Paxil, Zoloft, Celexa, Luvox. Many individuals who feel drawn to exploring experience with ayahuasca might be on prescription medication. Given the pharmacologic properties of ayahuasca, one has to be very cautious, however, about particular classes of medication one might be taking. And in particular, one needs to be very careful about adverse interactions between ayahuasca and the SSRIs, such as Prozac. Jace and I last year published an article in the Journal of Psychoactive Drugs describing what we consider to be potential adverse interactions between SSRIs and ayahuasca. The monoamine oxidase inhibiting properties of the hormonal alkaloids in ayahuasca impair metabolism of serotonin. If your system is pouring out more serotonin and preventing its reuptake because of antidepressants, you might be flooded with serotonin in your blood and in your central nervous system, leading to a very serious potential condition called the serotonin syndrome, which is associated with high temperature, severe tremor, confusion, possibly seizures. So one has to be very careful. I've heard of several individuals who unwittingly took ayahuasca while on SSRIs, had very frightening adverse reactions. One case study which we reported in our article was of a young man who, in the midst of a session, had to leave the session, staggered outside, collapsed on the ground, and for the next several hours was just in absolute agony with high temperature, shaking all over, terrible nausea beyond that which is normally encountered in the ayahuasca. But interestingly, he also reports that it was a very valuable experience for his development. No, seriously, what he said, he said he had a vision, and he hung in there. In that vision was, he said, I saw my wife, and I put myself in her shoes, and I was in her shoes when I was cheating on her, when I was having an affair, and I saw the pain that I had inflicted upon her. And he later credited that experience with what later turned out to be a healthy rapprochement with his wife. So ayahuasca, it's a mystery, works in many ways. I wouldn't advise this combination, however. So, caveat emptor, consumer beware. One really needs to be very cautious about potential adverse interactions. The work I've done with the approved work with MDMA has also pointed out the fact that individuals taking a variety of different entheogenic substances who are often deprived of accurate information because of problematic legal status of these substances often get themselves into severe difficulties and sometimes potentially life-threatening situations by taking entheogens or phenethylamines while also being on particular prescription medications. So be careful. What were our key findings? Just to sum up briefly, our UDV members were psychologically and physically healthy compared to our non-members. These men were robust, they were strong, they were healthy physically, psychologically, spiritually. There was no evidence of short or long-term toxicity from regular use of the tea. Many of the UDV members had life-transforming experiences on the tea, leading to positive and lasting life changes, and also the members attributed these positive changes to the combination of their tea and their context, meaning the context of the UDV. And finally, and very importantly, and really needs to be pursued with further investigation, regular users had unexpectedly high levels of serotonin receptors on their platelets. They had upregulation. And as Jace pointed out, we don't know of any other substance which could reliably induce this phenomena. And we believe that this might actually be a mechanism for a more efficient mood regulation medicine, where the receptors, being that there are more of them, are more efficiently responding to the serotonin available and in fact facilitating further secretion. So this is not a daily dosing regimen like we're accustomed to utilizing within the context of modern psychopharmacology. Rather, it is an occasional pulsing treatment. So, modern psychopharmacology might have something to learn from the ancient arts as well. And as I'm saying this, I'm remembering that in our hotel in Manaus, right across the street, there was a flashing neon sign that would flash in our room all night, and the sign said, "Drogas modernas." So we had the modern drugs flashing neon light at us all night, and we were there to study the ancient medicines, and I say medicine with a capital M. There's so much more work that needs to be done, and hopefully in the coming years we'll start to tackle this, and we're also very hopeful that we won't be the only group doing this work, that other individuals with scientific credentials and involved, and particularly individuals part of scientific institutions, will realize that this is a very important area that could provide for us some very important findings. But scanning, certainly the state-of-the-art brain imaging could certainly yield some fascinating results. There's a wonderful technology available in Finland. Actually, Finland, where Jace is at the University of Kuopio, has one of the more outstanding brain imaging units for serotonergic imaging, and we've got some prospective studies going there. acute modulation studies, additional pharmacokinetics you see here, studies with women. All of our subjects were men. We were trying to limit the variables, but clearly women should be studied as well. That would be a somewhat trickier study because you'd have to time it as in terms of when in the cycle you'd be studying your subject, but women should not be deprived of this kind of work. Post-mortem studies, further preclinical. Psychosocial. I'll tell you one study that I have that is up and running just recently in Brazil that I've been helping Glaucus de Souza Brito set up is a study assessing the status of adolescents who were exposed in utero to ayahuasca because their mothers were members of the UDV and determining, what is their functional status growing up. In the UDV, women take it throughout pregnancy. They take it in childbirth. They report to us that it is extremely salutary to their conditions. Babies are baptized. We went to a baptism. Babies are administered a little eyedropper full of ayahuasca. Intriguing experience. But then for almost all, there's virtually no participation of children in these ceremonies. When the kids hit mid to late adolescence, they have the option of attending or not attending. I was very interested in the functional status of these kids. I'm a child psychiatrist. I run a fairly large child psychiatry program, very interested in kids' development, influences on development. I'm very interested in the effects of drugs in utero. I'm part of a study now looking at the long-term effects of in utero of mothers who had abused methamphetamine. So this is an area that really, across the board, is a major area. So I was very interested in the functional status of these kids. And believe me, I've seen a lot of kids. I've worked up a lot of kids. These young people were very impressive in terms of their behavior, their personalities, their level of function. So just eyeballing them and living with them for several weeks as we did was somewhat reassuring. However, in Brazil, there are some political pressures on the church, and there has been a move to tighten up who can use the tea. Clearly, it's clear that the churches will be allowed to continue to function to administer ayahuasca within the context of structured religious ceremonies, but at issue is at what age could an individual initiate participation. So there's a question of whether or not the age will be raised to 21. The UDV feels passionately that ayahuasca, when available to the adolescents, is very good for them. That in fact they describe it as being prophylactic or preventative for getting into dangerous drugs of abuse. So they're concerned about this situation. So I've helped them set up a protocol which is now being implemented of assessing these teenagers in terms of their cognitive status, their psychosocial function, a number of different, I don't have time to get into it now, but hopefully if we have another meeting like this down the line, we'll have some data to present there as well. Other studies that should be looked at, clinical trials, there's a host of possibilities there. The one I'm particularly intrigued with is the putative role of ayahuasca within the context of treating individuals with severe addictions to drugs. As you all know, I'm sure that one of the most refractory conditions that individuals might be afflicted with is severe drug addiction. In fact, it's an area where modern medicine has not done very well. The most solidly accepted treatment today is the most solidly accepted treatment from 30, 40 years ago, which is 12-step programs. Interestingly, at the core of the 12-step program is the spiritual core to it. And, William James once said the best treatment for dipsomania is religiomania, and certainly perhaps one might imagine a model of ayahuasca-facilitated treatments inducing spiritual emergent phenomena, perhaps helping these individuals to overcome their life-destroying drug addiction. So tremendous opportunities there. Although, again, I would caution this is not a substance to be administered like conventional medication. One has to be extremely attentive to set and setting, extremely attentive to the structure, and ideally it should be incorporated within the context of religious ceremony. almost done here. These are our researchers and our institutions. Jace gave me this slide. However, he didn't include himself, his modesty, his humility. But Jace's name should be there too. These are our some of our publications. This one down here, just published in '99. It's anyone's interested, it's volume 65, pages 243 to 256, 1999, in the Journal of Ethnopharmacology. But we got our stuff published in some mainstream, high-level scientific and medical publications. This is our group. It consists of our researchers, our interpreter, and many of the subjects, the people in the UDV in Manaus. And here's an aerial view of the nucleocalpari in Manaus. So that concludes our discussion of the Huasca project. Let's take some questions, okay? I think we have time for a few questions. So if we can move the mic over into the center area. Okay, we're going to end at a quarter after. So do these work? Yeah. Okay, so Shirley, for you,
That's good.
a little shorter than usual. Please. You've only given this thing like 18, 20 times.
Did I what? Oh, you did your slide.
You'd think that by this time you'd have it figured out.
That's good.
okay, great.
You mentioned, hello, Sam? Hello? You mentioned biochemical measures of neuroendocrine studies, and you mentioned growth hormone, etc. I'm wondering, though, particularly in light of the information of the intrahepatic metabolism of the harmalines, if you did any studies on liver function tests and more mundane biochemical measures such as those.
Well, we did get baseline liver function tests, which turned out to be normal across the board with our subjects.
What else did you do besides liver function?
We did we did CBC. We did the full SMA panel. Also looked at renal function. I think we did a TSH. Right. Can't think of anything else. Yeah, there's all normal. No problems with anybody.
And you said you were going to comment on the ECGs that had deteriorated.
Oh, right. With a couple of the subjects, there was some bradycardia, slowing of heart rate. So the concern I had with individuals with perhaps some underlying cardiac vulnerability, there may be possibility of escape rhythms. I once observed one individual having a syncopal episode. It may have been related to that. I think individuals with histories of cardiac disease need to be more cautious.
But I'd like to comment on that. You have to remember that these people were sitting down. They were essentially in a meditative position. And a slow heart rate is not unusual for that. On the other hand, you look at the Santo Daime where they're dancing the whole time. So you're not going to see a low blood pressure in someone hopping around. I think this may have as much to do with the way the people were sitting during the experience than the drug itself.
Thank you.
Or medicine itself.
Okay, concerning the medical side effects, perhaps interesting, you noted some of the interactions, and perhaps now cardiac arrhythmias and so are also contraindication. The harmaline alkaloids can be a bit proconvulsive in animal experiments, so you could also imagine that perhaps patients with epilepsy should not be using ayahuasca.
Right. Individuals with epilepsy need to be cautious. It does
Exactly.
appear to have the proclivity to lower seizure threshold. We have reports on that. But let me mention something else. Jace's pharmacokinetic data is very important, and this is not only pertains to ayahuasca, it pertains to all drugs. We all metabolize drugs somewhat differently. Some of us are fast metabolizers, some of us are slow metabolizers. We're not all the same. Those of us who are slow metabolizers might be more vulnerable to side effects. In and of the drug itself alone or interactional between different medicines.
Can I just follow up again that it's quite characteristic that tremors are associated with high doses of ayahuasca. It's tremorogenic. These are characteristics of beta-carbolines, which the harmala alkaloids are. And That certainly will lower the seizure threshold. I would suggest anyone who's prone to seizures to stay away from these things.
Exactly.
Although, having said that, I'd like to jump in here and say, I mean, I think that all the safety concerns are important, and the points that Charlie's made about the SSRIs are important issues, and clearly anyone who's going to take ayahuasca regularly should be aware of these things. However, balanced against that, I don't think that there is any systematic screening of UDV subjects going on in Brazil. So 8,000 people or so are...
No,
Huh?
there is now. There
There
has
is
been lately.
now.
You know, we've
Okay.
talked quite a bit to Glaucus and some of the other physicians. They're now routinely screening for medications that people are taking for underlying medical conditions. And if they have concerns about medical conditions, they will not allow that person to participate with them. Keep in mind, this church is not into proselytizing. They're keeping their numbers small deliberately. The maestri that Dennis or Jay showed, who was giving our first subject his glass of ayahuasca, that maestri told me after 24 years as a maestri in the church, the Uniao de Vegetal, he has only invited in 6 people over 24 years. So they are deliberately keeping the numbers low, but they're also doing pretty thorough and I think quite responsible screening of individuals who do want to participate.
Well, I think the point is responsible screening. The point I was trying to make was that that considering the number of people who take ayahuasca on a regular basis, it's a remarkably safe medicine, and I don't think we want to overemphasize or exaggerate the safety issues associated with it, because then that will provide the authorities with a rationale to prohibit its use. So, we should be aware of the safety issues, but they should not be exaggerated.
Well, I think it's a very difficult situation at the moment. At least in Holland, we have worked together with the Santo Daime Church and make a long list of medical exclusions and interactions, and went through all the possible medical interactions you can think of, and made an informed consent based on that. I think all people working with ayahuasca in countries where there is debate whether it's a scheduled drug or not, and you do it like an organization, you can be sued basically if people get something nasty and they want to be nasty, they can be nasty. So it's a very difficult situation, and informed consent, where you really kind of make very clear what are the medical conditions you want to exclude, could be interesting, and could be important to kind of discuss on a cross-national basis what would be the major medical contraindications.
But it should be based on data and not speculation.
Sure, of course.
I'll add one more thing, is that it depends on dose too.
Mm-hmm.
And in my observation, the maestries are very careful in their dosing with people. Old people get less than young people, women get less than men, and certainly first-time people get a lot less than anybody else. So this comes only with experience.
Okay,
I
I had one preclinical remark on the 5-HT1A and alcoholism. I think it might be interesting for people to know that there has been studies with 5-HT1A agonists, where clearly rats, actually Finnish rats, which are alcohol dependent, they go down in their craving and their alcohol intake if you treat them with 5-HT1 agonists. So that's interesting, perhaps.
Which fits.
Exactly.
With all the good news I've heard today and the soporific effect of dark room and lunch, it's easy to forget that this is still taboo subject matter in our culture and theogenic research. I'm curious, do you think that an incremental accumulation of scientific studies like this will be sufficient to turn the tide of opinion, or whether something outside of science is going to be necessary to change our culture?
I'm hoping for divine intervention.
In the traditional diets, salt is mentioned as a taboo, yet I haven't seen any understanding of how that might reflect in reality. And I was also curious on the medical level if you know of anything about lithium carbonate, which is basically a salt, and clonopin, if it has any toxic reactions.
no idea on the latter, but the harmala alkaloids are involved in salt regulation, so I'm very interested in the effect of diet on these things. It's certainly real, there's no question about that. So lowering salt in a diet high in fish and bananas, which contain a lot of tryptamines, is quite interesting to me from a biochemical point of view.
Thank you. My question involves the oral activity of the Varola paste, and that was sort of mysterious because you're not getting the MAOIs in with it.
Right.
And it was said that there were some speculations as to the nature of that, and I was curious if you could share them.
Well, that's really all they are, is speculations that that there may be other components in the Varola paste that inhibit MAO. What I looked at was mainly the alkaloid fraction, and although the alkaloid fraction inhibits MAO, it's the tryptamines itself that are doing it. So the beta carbolines are not contributing to that. Or it may be that DMT is subject to other forms of metabolism, and it may be that there is an activation another way. It may have to do, for instance, with the matrix that it's in. The Varola pastes are full of tannins and lipids and this sort of thing. So the DMT may actually be, protected somewhat from inactivation in the gut and be able to be absorbed in an active form. But I wasn't able to follow up that work.
I did some follow-up on that.
Oh,
And,
well, I should have known.
We've been so busy.
Tell us, Jace.
We've been so busy we haven't had time to catch up on that. But
Right.
I think the beta carbolines that you saw in your analysis were probably artifacts from the preparation, and certainly not playing into MAO inhibition. But
Right.
I can't speak for the Varola because I haven't really gotten around to it yet. The Jurema is a Mimosa hostilis containing essentially DMT. Well, that DMT is not just DMT, it's attached to something. And I think that's carrying it into the central nervous system. So
Very likely it could
another
be the
unexplored
same
route,
sort of thing. Yeah.
totally unknown.
Yeah. But these more obscure entheogens are not going to get the attention that ayahuasca is getting because they're essentially, nobody uses them anymore. They're dying practices. So.
Can you comment on the alkaloidal content of Syrian rue, and if there's been any other studies on it, on other chemicals in it that might be harmful, or any other comments on Syrian rue?
Yeah, essentially it's harmaline, not so much harmine. It depends on the age of the seed, and there's no tetrahydroharmine to speak of.
Any studies on its long-term use or anything? Yeah,
And just
nothing
human use. It's been used for eye treatment for centuries in the Mideast.
And the Mimosa hostilis, it's DMT that's mostly in that? Is that the alkaloid?
Yes.
It's DMT.
Okay.
And there are other alkaloids in Syrian rue that are not beta carbolines.
That's right.
I don't think that their toxicology is very well understood. Jonathan Ott has suggested that you might be careful that perhaps by making aqueous extracts of the rue, you avoid ingesting some of these. So that might be a better approach rather than just taking the solid ground up seeds, because then these other alkaloids are there.
Thank you.
Good. Okay. That'll conclude this session. Thank you.